Dear students, today we will be talking about restriction endonuclease. So enzymes are the one which are used as one of the tool in biotechnology. You know that enzymes are biocatalyst. Once we know the type of reaction that is catalyzed by enzyme and its condition under which it catalyzes the reaction, we can manipulate them to be used as a tool in biotechnology. Now there are many enzymes that we are using. Some of the important enzyme are nuclease are exonuclease, endonuclease, and restriction endonuclease, which are one of the important enzymes. Then lysozyme, protease, lipase, DNA polymerase, reverse transcriptase, ligase, alkaline phosphatase. So we have already gone through these enzymes, how they are responsible for the reaction catalysis. We'll be discussing briefly about the restriction endonuclease. Now they are called molecular scissors as they can cut the DNA at specific site on DNA molecule. They are used to cut foreign DNA, vector DNA, and host DNA. The restriction enzymes are naturally present in bacteria, showing protection against virus. The bacteria produces restriction enzyme to break down viral DNA. Protection its own DNA means that bacteria, when it produces restriction endonuclease, then the virus is unable to affect the bacteria because it degrades the DNA of virus, and it protects its own DNA. How it is protecting? By doing the methylation of DNA. So the restriction endonuclease, they could not recognize the bacterial DNA, whereas the viral DNA which is present in the bacteria is degraded. So this is how the resistance against the virus is provided to bacteria. So this is how they occur naturally. Now in the biotechnology we are using it as a tool. So we have to isolate these restriction enzyme from the bacteria. To be used as tool in biotechnology, the restriction enzymes were isolated from bacteria. Now these scientists, Arbor, Nathans, and Smith, were awarded Nobel Prize in 1978 for the discovery of restriction endonuclease and their application to the problem of molecular genetics. And they discovered the first restriction endonuclease called EcoR1. Now let's see how these are named or how the nomenclature is done. So EcoRI is the restriction endonuclease enzyme that is isolated from E. coli. Okay, so the first letter, that is E, it is the name of genus. So Escherichia is the genus. Next two letters, that is CO, comes from the name of species. So coli, E. coli. R is the name of strain. So RY13, that is 13 strain, is the E. coli strain with which they were working. And Roman letter first is the order in which the enzymes they are discovered. So EcoR1 is the first restriction endonuclease that was discovered. Now let's find out how they act on the DNA. So they have a specific target site. The enzyme act on specific sequence on DNA and cut the DNA at specific point. These sequences are 4 to 6 nucleotide long and consist of inverted repeats. That is, if you read from right to left or left to right, the sequence of nucleotide will remain same in either strand of DNA. Such sequences are called palindrome. For example, let's in a simple form we'll discuss this word radar. So in radar, if you read from right to left, it will be RADAR. Okay, and if you read in the other direction, left to right, it will be again RADAR. So this is inverted repeat. Now let's see the target site for EcoRI. So One of these strand of DNA will have GAATTC, whereas the other strand will have its complementary strand, that is CTTAAG. Now let's see how to read this. Now why this is palindrome? It is because see the first one, 5 dash 2 3 dash GAATTC. Now the first one, if I read the same from 3 dash to 5 dash direction, then it will be CTTGAA. Okay, so second one, this one, 3 dash to 5 dash is CTTAAG, and if you read in the 5 dash to 3 dash direction, the same sequence, 3 dash to 5 dash and 5 dash, it will be GAACTT. So this is called inverted repeat, which occurs in both this, you can say, strand of DNA. So whenever such sequences are present, they are called palindrome, and the restriction endonucleases, they act on the palindromes. So EcoR1 bind to this sequence, both the strand, and it cut at specific site. So you can say between G and A, whatever phosphodiester bond is there. Similarly, if you see in the other strand, again between A and G, okay, so these are the site where it cut the DNA in both the strands. So EcoR1, produces sticky ends after the cleavage in each DNA strand has a little single strand portion at the end. So now you can see when it will cut and the strands are separated. So G is separated and AATTC is separated in 5 dash 3 dash, and if you see the other strand 3 dash 5 dash, CTTAA and G is separated because that was the site where EcoRI react. Okay, now they are separated. Okay, now these kind of and in which the one of the DNA strand has little single strand portion at the end, okay, they are called staggered end or cohesive end. Now certain restriction endonuclease, they produces blunt end like A3. Okay, so 5 dash to 3 dash, it will cut right after GG. So there is no sticky ends which are produced. So these are called blunt ends. So the mechanism, some of the enzymes produces blunt end, whereas some of the enzymes they produce sticky or cohesive end. Now just example of restriction endonuclease, Haemophilus influenza. From this it is the HIND2 restriction enzyme is isolated. Thermus aquaticus tag 1 restriction endonuclease is isolated. Now let's talk about another enzyme which is called ligase, and this is also one of the important enzyme. Naturally, this enzyme repair the double stranded breaks in DNA by forming the bond between 3-OH of one end and the 5-phosphate of another end. When you cut the two different DNA with the same restriction endonuclease, they will produce same end. Okay, like we have seen, it is going to produce, if I have vector DNA and foreign DNA, so the restriction enzyme will cut at a specific site. So in both the cases, what will happen? Same kind of ends are generated. Now let's see what is the role of ligase when you have such kind of two different DNA. So now when such kind of DNA are there, such DNA pieces are there, and when you cut, then you can see over here they will produce the same ends because the site is same between G and A in both the cases. Okay, now when we want to join them together, because see, vector DNA is present somewhere else and foreign DNA is isolated from somewhere else. Now in the test tube, what we are doing, we want to join the DNA. So for that we are using the ligase, and it utilizes ATP energy source. Okay, So when you ligate, then their whatever cohesive ends are there, they will join. Okay, and it produces this chimeric DNA. Okay, so ligase help in ligating the two pieces of DNA which are cut with the same. Now we'll be also discussing in brief certain other enzymes like endonuclease, which remove nucleotide from 5' and 3' end, that is the extreme ends. Endonuclease, now this is different endonuclease other than the restriction endonuclease, and they cut the DNA duplex inside the DNA at any point, but they will break one of these strand of DNA. Then DNA polymerase, which can synthesize complementary DNA on DNA template by using RNA primer. Reverse transcriptase, they can synthesize complementary DNA on RNA template. Lysozyme, it act as a it act on the cell wall of bacteria, especially on beta-1,4 linkage, which is present in peptidoglycan between N-acetyl muramic acid and N-acetyl glucosamine. Then different other enzymes like protease, lipase, which act on protein and lipids, are also used in biotechnology as the tool. So this is all about the enzyme, use of enzyme as a tool in biotechnology. Thank you.